Don't forget to subscribe to our channel and follow us on Twitter. And today we will have an exciting talk on computational reproducibility by Dr. Cetinkaya-Rundel, who is a faculty member at Edinburgh and Duke Universities and an instructor at RStudio. You may also know her from a popular MOOC on statistics and R. Dr. Cetinkaya-Rundel is also a strong supporter of open science, and she has kindly accepted our invitation to give a talk on reproducibility today, which is essential for all our works. Thank you very much for being with us today, and the stage is yours.
Thank you very much, and thanks for inviting me. So the name of the talk is The Results in Table 1 Don't Seem to Correspond to Those in Figure 2. And if you've ever worked on any sort of data analysis project, a paper where you did summarize things this way, it's possible you encountered this at some point. And the background picture here, it's a little pixelated intentionally, but really that's this picture. So I think this could have been a better opener for the talk itself. It is an utterly frustrating situation to find yourself in when this is the case, and it can happen so easily. So in this talk, we're going to talk a little bit about how these things happen, how we end up with, even if you're doing meticulous data analysis. So we're not just talking about humans making mistakes here. It is more talking about the tooling and the infrastructure necessary so the humans don't have to think about trying to match up results in table 1 with those in figure 2. So I'm going to start with a set of random numbers. And so I've picked a set of random numbers, and I've done so in R. And I want to show you a quick bit about how we can actually come up with the same set of random numbers. So this is just one quick tip. I'm randomly generating numbers, but before I do so, I'm setting a seed. A lot of us do work that involves simulation, either within a modeling context or in an optimization context, and simulations are essential. They do need to be random, but at the same time, they need to be reproducible as well. So this is one quick tip for doing so in R. Whatever computational language you're using probably has something similar to go along with it. So whenever you're doing a simulation, or even if you personally might not be doing the simulation yourself, but you might be using a package or an analysis tool that actually does some simulation under the hood, make sure that you set a seed so you can actually reproduce your results. But let's throw around a few other numbers that are not actually randomly selected. So 70, that's more than 70% of scientists have tried and failed to reproduce another scientist's experiment. This is from a survey that was published in Nature News in 2016. And it was a survey on 1,500 scientists, and they asked them these questions about, like, have you tried and failed to reproduce another scientist's experiments? So that's a striking number. 50, that's more than 50% have tried and failed to reproduce their own experiments. And that is where we're talking about. This is not about somebody else not taking meticulous notes. This is about your workflow not even allowing yourself to easily reproduce your own work. 152, that's in Google Scholar yields 152 results. This is as of March this year, containing the term reproducibility crisis just in 2019. So the idea of reproducibility crisis is upon us, and the number of scholarly publications about them is even increasing. And there's also a lot of mentions of this in the popular media as well. 1992 is the earliest reference in reproducibility research that I could find. So this is a paper called Electronic Documents Give Reproducible Research a New Meaning. And we're going to take a little bit of a look at that. But I want to draw your attention to this 1992. It's been quite a few years since then, and we're still kind of battling the same problems and also finding wonderful solutions to them as well. And technology is evolving in a way that's helping scientists do their work reproducibly without having to do a whole lot of extra work. So this is the paper from 1992, and I want to focus on one snippet of that first. So these two scientists from Stanford said in 1990, they set a sequence of goals. I'm going to quickly summarize those for you. Learn how to merge a publication with its underlying computational analysis. Teach researchers how to prepare a document in a form where they themselves can reproduce their own research results a year or more later by pressing a single button. Learn how to leave finished work in a condition where coworkers can reproduce the calculation, including the final illustration by pressing a button in its caption. Prepare a complete copy of our local software environment so that graduating students can take their work away with them to other sites, press a button, and reproduce their Stanford work. This is where the research was published. Merge electronic documents written by multiple authors. Export electronic documents to numerous other sites so that they can readily reproduce a substantial portion of our Stanford research. Now think back to your own research, the projects that you work on, and think about how many of these bullet points can you readily check. I think some of these technologies are out there. This idea of being able to press a single button to reproduce your work, I'll be talking a little bit about R Markdown in a little bit. And this is, at this point, a solved problem. It was a solved problem many years ago as well. If you're a LaTeX and Sweave user, you know what I mean when it comes to literal computing. It used to be technically a little bit harder to do so, or at least had a steeper learning curve, but I think these are solved. At the same time, the learn how to leave finished work in a condition where coworkers can reproduce the calculation, including the final illustration by pressing a button in its caption. I don't think many of us can check that button very easily, even today, more than 20 years after the publication of this report. So they said they actually have managed to fulfill all those goals. And now that they have begun using CD-ROM publication, so think about when those were a thing, we can go much further, and every figure caption contains a push button that jumps to the appropriate science directory and initiates a figure rebuild command, and then displays the figure, possibly as a movie or an interactive program. This basically got accomplished finally in 2018. So this is one of the first examples of a live code article. Since the publication of this one replication study, there's been more of these out there. They're a little bit of a proof of concepts still. The experience, I wouldn't say, is wholly natural, and it can be a little bit laggy. But that being said, and you can see the link at the bottom, any of these references that I'm referring to at the end of the talk, I'll give a link to a GitHub repository that actually has these papers and links and everything. So you can actually go and play with them yourself. But this particular publication actually has a reproducible versions of its figures. So if you click on the blue circle on the screen that says R script, you would be actually launching a live R session with the data loaded, and you could reproduce this figure and you could play around with it as well. And so here we're talking about push button reproducibility. That is, as I said, not the smoothest experience just yet, but something that was set forth in that 1992 article, and we have accomplished it. And I think there's still a long way to go before any scientist publishing a paper is able to say, hey, I can do this. And I want to take a moment to say that this is not just a scientists aren't doing their work reproducibility problem. Journals aren't necessarily equipped to host these either. So there's work that needs to happen on the scientist end, but there's absolutely work that needs to happen on the scientific publication infrastructure end for all of this to be feasible and all of this to be able to be done without additional expenses having to be incurred by the researchers. So let's set the stage real quick. We're going to be talking about reproducibility, but I want to make a distinction between replicability and reproducibility. In both of these situations, we're talking about the same research question. We want to say that if a study is replicable or reproducible, you should be obtaining the same results. When we talk about replicability, the way I'm defining it here is that you're using a new data set and you're getting to the same result. So you can think about a medical experiment, for example, that was done on a certain sample from one population, particular research question, and some results were obtained. Now, what if we were to replicate that experiment on a new sample and see if we get the same results? That would be, if the answer is yes, we would say that study is replicable. When we're talking about reproducibility, I'm defining it as same research question, same results, and same data. So I worked on a study, I collected some data and have analyzed the data and come up with some results. Now I pass on my data set to you, and hopefully my code and whatnot to you as well. Are you able to actually receive the same results? it seems like it should be a no-brainer that you should be able to. We're starting with the same data set. You have my code for the analysis. It is unfortunately not necessarily a no-brainer, especially if some time has passed in between, especially if we're using different computing environments. So there are a lot of factors that actually still go into being able to receive the same results, some of which are about how I've organized my work as a scientist, and some of which are things about your computational infrastructure as well. So this is the reproducibility that we're going to be talking about today. There is a lot being said about replicability as well. So the American Statistical Association over the last few years has had some publications about their statement on p-values. So making statistical decisions based on p-values tend to sometimes yield to results that may not be as replicable, because perhaps they're not the best way to be making decisions about our research questions. There's also another article here, and as I said, these are all in the references folder in the GitHub repo I will be pointing to at the end of the talk, called Why Most Published Research Findings Are False. It sounds like a BuzzFeed article. It is a little buzzy, it's a little clickbait, but indeed it's a really, really good read in terms of why this is the case. These are very important questions other people are working on and talking about, but again, we're going to be talking about the computational reproducibility aspect of things today. So let's give a quick example. If you've ever worked in R, you've probably come across this data set called the IRIS data set. Even if you haven't worked in R, you probably have seen examples using this IRIS data set. And so I'm going to do that. And the reason why I've picked this is I'm thinking that this is a data set at least some of the audience is familiar with. So I have a regression output for predicting petal length from sepal width of these flowers. And as you can see, I have this regression output here, and I have a regression coefficient for it as well. And here is the same relationship visualized, the relationship between petal length and sepal width. Take a look at this real quick. Do you see any mismatch between my results in Table 1 and those in Figure 2? And I'll give you a hint. The regression coefficient is negative in Table 1. But I actually have a positive value here in Figure 2. How did this happen? You might be thinking, well, you just made a mistake, but really, let's think about how might this happen in a paper. So let's say that I'm doing my analysis, I write my report about this IRIS data set a little bit, and then I actually go ahead and fit a model where I predict petal length from sepal width, and then I print out the model summary. And the way I print it out into my report is I run my code in R, I copy and paste it into my report that maybe I'm writing in a Word document or a Google Doc or something like that. Then I go ahead and keep doing a little bit more writing, explaining my model output. Then I say, okay, now is the time to provide visualization as well. And I go ahead and actually visualize the relationship between these two variables, copy and paste the visualization, put it into my report, and you can see that I have the negative slope and the negative regression line here. Now, if you're familiar with this data set, you'll recognize that there are these 3 species of data that are included in this data set. And actually, the reason why these 3 species are included was originally the point of the collection of the data was to show that 2 are similar to each other, and the third one is not, the setosas are not. But if I'm doing this analysis and realize that, hey, that one species is completely off to the other side, I might decide, you know what, I'm going to refactor my work here. I'm going to actually remove one of the groups and only do the analysis on the other 2 groups. So then what I do is I actually filter out the setosas, copy and paste, and say, okay, this looks much better. This is the work I want to be working on. And then I go on my merry way. Well, what happened is I completely forgot about the table that was up top, and I completely forgot to change it. And if this is a realistic report, those are probably not even happening on the same page. That's probably something happening a few pages behind in my report, and it is an incredibly natural human error to forget to change that up. And this is how you might end up in a situation where your results in Table 1 does not match up with those in your Figure 2. The thing here is that I was relying on myself, a human, to remember to go back and make the changes. And this is the point of failure in this analysis. And this is how I'm ending up with results that don't really match up and that might be hard to reproduce. The copy-paste bit is the bit that we basically want to remove from this paradigm. So how do we easily do that? How do we make research more reproducible? Here's a quote from David Donahoe, who's paraphrasing another scientist, which says, an article about computational science in a scientific publication is not the scholarship itself, it's merely advertising of the scholarship. The actual scholarship is the complete software development environment and the complete set of instructions which generate these figures. So how do we do this? We want to make our raw data, our code and documentation to reproduce the analysis, and the specifications of your computational environment available and accessible if you actually want to say that your entire research and publication is a reproducible publication. How do we go about doing that? This is another quote I really like from Keith Baggerly, who's a scientist at MD Anderson who does a lot of reproducibility studies. The most important tool is the mindset when starting that the end product will be reproducible. So you can potentially achieve backwards reproducibility, meaning that look back on a project that you have already completed and say, let me organize my files and folders now that I'm done with my research and now that the paper is in publication so that someone else may be able to reproduce it. Or maybe even the journal you're applying to is now necessitating that there be a reproducibility component to your research and are asking you for your files, and that's the time that you're trying to make your work reproducible. I don't think that's a good approach. There's a good chance you'll fail at it. And even if you don't, even if you do manage to succeed, it's not going to be painless. The other thing is that currently our academic structures don't necessarily reward the time you would spend on organizing your files after your publication for reproducibility. So now you might be thinking you're doing this chore, if you will, after your work is quote unquote done. So instead, you want to be thinking about reproducibility from the outset and start with a mindset thinking that how do I start this project so that I keep reproducibility in mind along the way. And when I'm done with it, I am confident that my work is reproducible. When we say reproducibility, let's think about where we are in terms of the kind of the grand scheme of reproducibility. On one end, you have nobody, not even yourself, can recreate any part of your analysis, which is basically a mess. On the complete other end, you have push-button reproducibility in published work, kind of like the paper that I was showing you is a proof of concept. Maybe you aim for somewhere here. The push-button reproducibility may be impossible or difficult to achieve for you, and there may be a variety of reasons for doing that. Some of the most common reasons I hear are the data you work with are private, so you won't be able to share that. Well, then you can't get push-button reproducibility of every single figure in your paper. Another reason might be that your data set is quite large and there's not currently a good way of kind of bundling all of that analysis up and sharing it with another researcher who can reproduce it in a secure way, for example. So there may be these sorts of reasons why it's not happening, but you want to get as close to it as possible. And when I say as close to it as possible, it doesn't necessarily have to be that you're hitting our target. Somewhere on the higher end of this spectrum is probably good enough. And the important thing is with every project that you start, you want to look back at the previous one you've worked on and think, well, where was I in terms of reproducibility there? How can I take one more step forward? There is no one-size-fits-all solution for computational reproducibility, and this is somewhat stemming from some of the issues we discussed earlier in terms of data privacy issues, data size issues, computational requirements as well. But you need to think about what are some things that you can do regardless of the subdiscipline you work in that might be helpful for you. So I'm going to be talking about 8 principles for computational reproducibility that might help regardless of the type of specific work you're doing. The first is organization. So organize your project. What do I mean by that? If the spectrum of level of organization is somewhere between this happening on your desktop to complete Marie Kondo level organization, again, you want to aim for somewhere around here on the higher end of the spectrum. How can you do that? If you have a simple analysis that you're working on, have some subfolders. Every single project that you're thinking of should be a folder on your computer. And within that folder, you have a folder called raw data. That is where the wholly raw data goes in, and it never, ever gets touched. And by never gets touched, what I basically mean is that you never double click on that CSV file to open it in Excel, for example. You don't want to be interacting with that file. And in fact, if possible, you want to leaving it in only a read-only format and then using a scripting language to do anything with it. Chances are the raw data that you are starting with is not going to be your analysis data. Maybe you need to figure out, clean up some headers. Maybe it's a spreadsheet that has some junk on top and at the bottom that you need to strip out. Maybe it has too many columns that you don't need and is kind of taking up memory when you're working with it, when you're doing your analysis with it, and you need to trim it out a little bit. Save the raw data as is in, if possible, a read-only format. Have your manuscript as an R Markdown document that actually uses the raw data and saves the process data as well along the way. And then the process data is the data set that you're using for your, for example, modeling or simulations. In a simple analysis, this might work. A single R Markdown document might work. But if you're thinking about a more complex analysis, then you might actually want to have your computational steps not all embedded in your Markdown manuscript, but instead saved as scripts that go into a scripts folder that actually perhaps produce some figures that get saved out. And then your R Markdown document is where you have your prose and some of your code, and that also pulls in some of these figures. And remember that if you're working in R Markdown, those code chunks that you have might be bare R code, or it might be chunks of code that actually source those scripts. In terms of the internal organization of this project folder, the examples here are one type of example for doing it. It may not be, as I said, there's no one size fits all reproducibility, so it may not necessarily work for your subdiscipline. So stick with the conventions of your peers so that if you were to share this folder with another colleague, they actually might have an idea in terms of where things might be saved. Number 2, write readmes liberally. So if you have raw data, here I've taken an example from the New York City flight data set in the dplyr package in R, and it comes with a few flat files. So I've saved each one of those as a CSV file here, but I also have a README that goes with them. This README actually tells me where these data come from, and it tells me what else is the contents of that folder. This is going to feel redundant initially, especially if you're working by yourself on a project and you're not even sharing it with anybody else, and you might be thinking, I know what's in these data files and they're named so beautifully as well. But chances are, a few months down the line, you're going to forget what you were working on because you'll have moved on to another project, and it's really, really nice to have these notes for yourself that you can come back to. Number 3, keep your data tidy and machine readable. So what do I mean by this? I'm going to give you an example from a type of a file I interact with regularly, which is student grades. Okay, so let's say I have these 10 students, right? Yeah, 10 students in my class. I give 2 exams to them, let's say. So I've recorded their exam grades, and I also know their majors as well. But what I've done here is there's some of the majors that are unknown. So Gabrielle Cherry, the person at row 5, I don't know what their major is, so there's a dot there. So one person was sick for one of the exams, another one missed it. Is there a difference between these two? I don't know. And I've also used coloring to say these are students I kind of have my eye on. They haven't been participating much. Maybe I want to check in on them. Well, this type of data set, when a human looks at it, it's digestible, but note that there are only 10 observations here. If I had many, many more people here scrolling through a spreadsheet with lots of students, and with this sort of coloring is not going to really get me far. So what I need to do is I need to turn this into a machine readable format, which means that I want to record the code and document the non-code steps and write some tests for going from one to the other. So what are some code steps that I've done here? I've actually split that major column into first and second major so that they don't need to be treated as text strings, but I can actually use them as categorical variables as well. I've replaced all the different ways that I've said some information is not available with an NA, so those can be excluded from certain sorts of analyses. And then I've done one manual step as well. I've actually, as a human, gone through the coloring of the rows and coded people in a new column called participation is okay or low. So now this particular data set is machine readable, and I can tabulate things. I can calculate things like what percent of the students are doing low participation, what is the average exam 1 or exam 2 score for people who have low participation or not, so on and so forth. Prior to that, I could do those by hand, or I would need to have particular code steps for getting through that process. But by making sure that my input data is machine readable, I'm able to move on with the analysis a lot quicker. Number 4 is comment your code. And what do I mean by commenting the code? So here I have that same plot that we were looking at earlier from the iris data set with the 2 species. But what I've done is instead of fit a linear model here, I've actually applied a smoothing. And you can see that the method for the smoothing is lowest. And I've also used a span argument for 0.375. So that argument tells me something about how smooth that fitted curve is going to be. Why 0.375? So yes, I have some comment on top of that code chunk that says use lowest smoothing, but I haven't said why use lowest as opposed to something else. And I haven't said why I picked that particular parameter. These are the types of things you want to be commenting as well, not just the what. The what is obvious from the code. It's not always obvious because your code may not always be this simple. So sometimes it is useful to explain the what, especially if it's going to take a lot of cognitive load to figure back out what you were trying to do. But in addition to the what, which is the easy bit, you want to make sure that you're recording the why as well. Sometimes the why might be a lengthy explanation. So maybe in your code comments, you put a link to something else. You put a reference to a paper or something. And sometimes it's just, I tried a few things and this is what I settled on. Do not count on your memory to remember why you picked a particular smoothing parameter. Make sure that you document that step. It's possible you won't remember it, or it's possible you'll pass this on to another colleague or a collaborator and they'll be thinking, why this number and not 0.5 or 0.1? Number 5, use literate programming. So we talked about R Markdown earlier, and this is where it's going to shine through. So this is an R Markdown document. And you can see that in my R Markdown document, I have on top what I call a YAML that's happening between lines 1 and 8, fenced with the 3 dashes that actually has some meta information about my document. So a title field and an author field, and that you can see that I can actually embed some R code into it. So that's going to show me what the date that the document was compiled is in. But then if you scroll further down, you'll see that there are R code chunks that are shown in slightly gray. And then I also have some prose in there as well. So lines 21 and 22, for example, are giving me some text that are actually maybe my interpretation or my introduction or my data analysis. This is what the result of this document looks like. You can see that the code is there, but in addition to the code, I also have the output as well. So at the bottom of that image, under the model section, not only are you seeing the code to fit a model, but you are also seeing the R output. And I have not copied and pasted that output from R in there, it basically compiles to that document, which means that your code and your output are completely inseparable. And so if you look further up in the document, if you're looking on the left-hand side, that's happening between line numbers 24 and 27. We have an R code chunk that filters out the setosa species, which was something I did on my kind of mock demo earlier, where I said, how would you end up with a mistake where your figure and your table outputs don't match? Here, what we've done is we've actually done the filtering up top in the document, and the entire rest of the document now uses this iris non-setosa data set, which means that mistake I made in terms of not remembering to fix up my earlier table is simply not going to happen here. There's not room for that human error anymore, because the code is being compiled every time you knit your document. You can also think about a situation where you like this paradigm, but your audience who you're going to be sharing this report with does not need to see the code. That doesn't mean you should have to take the code out of there. So simply by changing a parameter in your document that tells you whether the code should be echoed, in other words, printed in your output or not, and you can do this at the document level like I've done here, or at chunk levels, you can go from a source document that has the code to a final output that doesn't show the code, but only the results. If you're working with collaborators who are also read R code, this is probably what you want to share. If you're writing a scientific paper and you're going to be trying to get it ready for publication, this is probably what you want to be looking at, because you're not going to select the code visible in the printed output, but it should be in your source document. Number 6, use version control. So one of the things that we're doing in our Markdown is we're putting our code and our prose in one document, but we're making changes liberally along the way. So what you want to be doing is you actually want to be tracking your changes using a version control system. And I'm going to be talking about Git here. There are other version control systems, but the reason why I'm highlighting Git is its facility for using it with RStudio, using the RStudio IDE. So if you are already coding in R, this is probably a good way to go. And then you can host this stuff on GitHub as well. I'm not going to go into the details of how to set up version control, but I do want to point your attention to a incredibly valuable resource called Happy Git and GitHub for the user. This is from Jenny Bryan and a few others who have contributed to it at happygitwithr.com, and it's basically a resource for our users to be using Git. Git is a version control system that was originally developed for a lot more than just doing data science and statistical analysis version control. It is actually used widely by software engineers, and there's a lot of capabilities to it that can get overwhelming. If you are starting with version control now, what I would recommend is to find a resource like this. If you're an R user, this is the right resource. If you use a different language, find a resource similar to this that has been designed for using Git for the types of tasks that you do, whether that be data science or software engineering or what else, as opposed to kind of trying to learn the whole thing, which is going to be overwhelming. Number 7, you want to automate your process. So we looked at something like this earlier. We said we have some scripts. I like numbering my scripts so it's very clear in which order they should be ran. That being said, I'm relying on a human, myself, to run those scripts in that order. It's probably a low-cost way of doing this would be to add another script at the bottom that actually has something like this that says run all of the code by simply sourcing these files in order. So instead of you as the human remembering to run them in order, you're relying on another script to do things in order. You can go beyond this, of course. So that was a low-cost kind of way of doing it simply within the R framework. A slightly steeper learning curve, but one that would have a good payoff would be to learn something like make. And so I have linked here to a kind of a short tutorial for using make in the context of writing a scientific paper where the analysis is done in R and the paper is written in LaTeX from Carl Broman. It's a really good resource for kind of getting started with make. And basically what it is, the snapshot you're seeing in the middle of the screen here on the slide is a plain text file that basically tells make in which order to run things. And so if you've ever done paper writing with LaTeX that had citations, you know, you need to LaTeX and then BibTeX and then LaTeX and then LaTeX again in order to get all your citations working. That's what those 4 lines of code are doing, actually running either PDF LaTeX or BibTeX on your LaTeX in your tech files. So the idea is to think about what are the human steps that would be required to reproduce this, and then actually writing them down using this particular syntax for make, and then asking the computer to do that for you. The last tip that I'll mention is to share your computing environment. And this is something that is hard, but is becoming easier with many incredibly smart folks with resources working on this. There are a variety of approaches for doing this. If you've ever heard about Docker or Docker containers, that's one way of kind of creating a computing environment that is containerized, as in it perhaps you're developing it, but it's not necessarily, it's being built as a virtual environment that somebody else can take and recreate as well. Another one is Binder. This is a newer project. You can actually add this Binder component to GitHub repositories, and it works well for open source projects. If you have some R analyses that you have a bunch of scripts for, what you can do is you can launch a Binder session and kind of run them there. So somebody else arriving at your GitHub repository, instead of downloading your files and running them on their computing environment with whatever local setup they have, they're going to be basically coming into the computer environment that you have set for them. It is slow, but you're launching a new completely new session there, and that's why it's slow. It's also kind of unclear with Binder, at least as of today, how it would be maintained, because if you're not paying for the compute time, then who is? And if it's completely free, is it going to be free forever? But nonetheless, I think it's a wonderful advancement, and it's one that's a lot easier to learn and use than learning about Docker and setting up those containers. Another option that I would recommend that actually requires a lot less technical chops and has limitations that comes along with it, but one where you can simply have someone else drop into your R session would be using RStudio Cloud. So this is a hosted service by RStudio where you're basically launching an RStudio session. So you can put your files, your data analysis, whatever, if they are okay to be uploaded to somebody else's server, and simply share that project with a collaborator. That could be for a complete project. It might be for a bit of a thing, or perhaps you're going back and forth in email saying, yeah, but I'm running this and I'm getting that, but I'm running this and I'm getting that. You can simply share your computing environment that way. So these are the 8 tips that we talked about. There may be different ways. I've given at least one example for each one of them in terms of how you can implement it, but there may be different ways that work better for your self-discipline. But at a minimum, I think if you're thinking about these 8 tips, and not necessarily, if you're not doing any of them just yet, don't think about, okay, next project, I'm going to try to do all 8. But maybe try them, points 1, 2, and 3 for the next project, and then you can work on 4 for the other one. And just, as I said, try to do one better each time you're working on a new project. There's a really nice paper called Good Enough Practices in Scientific Computing. It's actually a follow-on to a paper called Best Practices. Sometimes we thrive for the best, but good enough might just be good enough. And I think it's an encouraging paper in the sense that it gives these concrete tips in more detail than I've described, and further tips as well. And it basically talks about how it can simply be doable at some level. Now, looking into the future, I want to wrap up by talking a little bit about how we prepare scientists for the future as well. So I showed this quote before, which said the most important tool is the mindset when starting that the end product will be reproducible. And here's a follow-on quote from Carl Broman. The second most important tool is indeed training. This webinar serves the purpose of some training at the level of exposure to different tooling that you might want to adopt for making your projects more reproducible. But that's maybe at the level of number one. So I want to talk about this kind of two-pronged approach for training people. One is to convince existing data scientists to adopt a reproducible data analysis workflow, and this is a hard one. If they don't already do that, to ask people to change their ways is always hard. Number two is another thing that I work on as part of my faculty position. That is not an easy job, but at times easier, is to train new data scientists who don't have any other workflow. And what this means is that you're not trying to break their existing workflow, you're simply telling them this is the only way to do things. So as a statistics and data science educator who teaches data analysis, I feel like one of my responsibilities should be to instill best practices in students before they set out to do research. So that is my piece of the pie. And the way we do this in the courses that I teach at the introductory level is students access a computing environment in the cloud, so in their browser, as opposed to local installations. So we know that everybody has the same setup. They authenticate, and they find themselves in a fully functional RStudio session, either using RStudio Cloud or another hosted server system. Every single thing they do, every single assignment, every single report is in our Markdown document. So that whole copy-paste paradigm never enters into their workflow ever. And that our Markdown document looks something like this. So this is an example of a data set I use on the first day of the class, where students actually recreate this analysis. Now, when I say recreate, they don't actually write all of that code on day one of the class. That would be crazy. But we're actually able to share that fully functional R Markdown document with them, and they simply change a few parameters out to see what a reproducible workflow does look like. And then they do some interpretations on the plots that they see. It's about voting patterns of various countries in the United Nations General Assembly. And then we break apart this example over the next weeks where they actually learn the code itself as well. And everything that they do is then submitted is version tracked with Git and then submitted as a GitHub repository. So instead of using a learning management system like Blackboard or Moodle or Sakai, we actually use GitHub for students to get their assignments and return their assignments, which means they get used to the process of working with Git in the context of a data analysis. And they have to kind of get used to it because that's what they need to do in order to get graded on their work. So that's kind of all I had for this. I have two links here. The first one will take you to a slideshow that actually has the slides that I went through in PDF format. And the other one will take you to a GitHub repository that has the scripts for the figures that were used in this talk, as well as the papers that were referenced. And the links are live there as well. So anything that I linked out to, you should be able to click on and either access it through the GitHub repository or get to it on the web if you would like to read further about them. And I hope that the talk has been kind of inspiring for taking your reproducibility a step further. And as I said, one size does not fit all, so it's possible some of the things you can see how you can exactly adopt tomorrow, and others you're scratching your head thinking, I really don't think that would work for me. But the mere fact that you're thinking about it and potentially trying to come up with an alternative that would work with the type of data that you work with or the analysis that you work on is still a step forward. And I'd be happy to answer any questions. Thank you.
Thank you very much for this interesting and extremely helpful talk. We have some time for questions. If you can use the chat box, I will be reading your questions. Maybe while the questions are being written, I can start with one. The most common problem I encounter is actually at least my research actually takes a long time. And what happens is all the databases get updated, all the sources I use get updated on the way. And it's like the initial stages use another version and the latest stages use another version. And it's not really possible to just rerun everything at the end because it takes a long time. Do you have any suggestion for these kind of problems?
So I think what you're describing is a good candidate for actually using a version control like Git, where you are actually able to do track where things changed, because if the data gets updated, you're maybe able to have some time points or commit messages that signify that. Another thing you can do with a GitHub repository, if this is relevant, is have releases along the way. But basically all they do is they zip up everything that you have up until that point and save that version, and then you can continue to make changes to it. So I think organization is going to be key, and a version control system, as opposed to thinking about, the simple example I gave of an R Markdown document where you're constantly rerunning your analysis every time you knit it. Instead, you want to be keeping track of changes using version control, and have the raw data untouched, as I mentioned before, and any sort of figures or results that you generate are saved in their individual folders with particular commit messages that are going to be time-stamped anyway. So this will help you kind of decipher where things came from ultimately at the end. And if you are writing a research publication, that itself has to be reproducible. So how do we make that reproducible? Maybe interim model objects like model outputs and stuff get saved to other files, and the paper is simply reading those. So you're not having to rerun your models, but you are at least having to rerun the code that parses that model output and maybe makes a summary figure or something like that for your paper.
Okay, thank you. Thank you for the suggestions. So there is a more like a comment, I guess, thanking. Thanks for the suggestion of Wilson paper. Also, I was always trying to do all things in once, but I think, like you said, it's important to be better from the previous project. They said. Any questions or any other comments?
No?
Yeah, I think there isn't any question, so
Okay.
maybe, yeah. Okay, thank you very much for the talk.
You're welcome. Thanks for having me, and hopefully, people try to do one better next time they're working on a project.
Yes, exactly. Thank you very much.
Bye.
Bye.